the interest of time. So I have a very initial question for all of you, which is something you should have all asked yourselves before I do. But how many different codon combinations are there in the human genome? And what is a codon? Define that for us first. Okay, sheet up. These are 3 base pairs. How many possibilities can we have in the first position? What are the different bases we can have? 4. Abhishek, how many different bases can you have in the second position? Tashvia in the third. So who can do the quick maths? What's 4 times 4 times 4? Okay, 64 different codons. Now, Alicia, what does a codon code for? Revise.
It codes for a specific protein.
Let's zoom out, everyone. What process am I talking about here? Why should you care about codons? Good. So you should straight away shift your mind to photosynthesis. Now, which part am I talking about? Transcription, where I'm converting. Let's pause for a moment. Transcription, where I'm converting your DNA to RNA, or translation, where I'm converting RNA to amino acids. Translation, the latter. So what does each codon code for? When it's sitting on the ribosome, and when that ribosome is reading a set of 3 bases, what is it going to recruit? tRNA molecule. And what's in the back of that tRNA molecule? Good, right? The answer is amino acid. Now, how many different amino acids can you have? Now, you tell me this. If there are 64 different codon combinations, coding for 20 different amino acids, what does that mean? What is the implication of this? One amino acid has multiple different codons. But this is a question. Chicken or egg, which came first, the codon or the amino acid? The codon, because that was sitting on the mRNA. So I think we should flip that statement to multiple different codons code for the same amino acid. My question is why? The big question you should always be asking yourself when you're learning, right? Why is that the case? Any of the online students? Georgina? Lookman, what do you think?
Because many, actually I don't know, I'm not sure.
Sushita? Any ideas? Why is this the case? Did everyone hear that? Did you all get that? Tanisha? What she said? No? She said something very important, so I want you all to pay attention. Get a bit louder for us.
If some of them, if one of the bases changes, it might not affect the amino acids and can have the same amino acids.
Good. Let's have a thought experiment, right? Imagine you're sitting in Bondi Beach. Finish your exams, really happy, peak summer. It's middle of the day. How many changes to your bases do you think would occur in 1 hour? Pick a number. Ashlia? 20,000. 20,000 letters will randomly invert from a T to an A or a G to a C. So that's how many mutations you accumulate in 1 hour in the sun. Now imagine if you have this phenomena where even if you change from an A to a T, that codon will still code for a leucine, or it will still code for a protein. You see how this is a protective feature of DNA. This is what we call the redundancy of the DNA sequence. Redundancy of the DNA sequence. What are some other terms we use to describe DNA and DNA replication? We use the word semi-conservative. What does that mean, Prithvi?
It has its original strand and it has a new.
Every time DNA replicates, right? It's split into 2, and then a new strand attaches to that complement. Okay, so that's semi-conservative. What does anti-parallel mean?
They run fractions, 5 times 3 times.
Good, right? So one strand will run 5' to 3', which refers to the bonding of the sugar and phosphate, right? And the other strand is parallel, as you see. It's kind of like a train track, right? Which kind of twists on itself, but it runs the opposite direction, so it's anti-parallel. So this is another word you use to describe DNA. Now, the reason I brought this up was you get questions like this in your HSC, where they give you a statement or a phenomenon, and you have to discuss or explain or justify that. Does that make sense? Good. In this case, we're going to now start moving on to this dot point here, 3.3. So you can all get to that. It'll say page 5, which is a bit funky, but that's where you should all be at. Good. Now, who learns about Gregor Mendel at school? Put your hand up. No? At all? Okay, well, I'll tell you guys a little bit about it. When I see scientists, I want to know what they look like. His names otherwise are just boring, right? Does this guy look like a scientist to you? He wasn't a scientist. He was actually an Austrian monk. But he's been immortalized in biology because he was just curious, and he led to 3 principles that are now the laws of cell biology. 3 principles. Now, I want you all to tell me, if a tall person married a short person, what would you expect the offspring to be? Tall, short, middle height? Think about your friends, don't just jump straight to an answer. Think with observations around you. What's a general principle you note? Any ideas? That's it? You think they'd be middle, right? And that's what people thought in olden times. We didn't know what DNA was. We thought that there was a blending of characteristics. So at Mendel's time, in the 1800s, we thought that, when a tall person married a short person, or blue eyes and brown eyes, you got a blended variation of both traits. Mendel wanted to test that. Now, is testing humans complex or easy? Yeah, look how long we live, 80 years. It takes 9 months for a baby to go from conception to birth. There's diseases. So what he used was a simpler model. He used plants. Why would he have used pea plants? What's the benefits? Think about it. If you were a scientist, why would you not pick 100 humans, and why would you rather grow 100 pea plants to test genetics? Ethics, major reason. Very good. What else? Yeah, way easier. Shorter generation times. There's no chemistry, attraction, all of that. Just pea plants, make a baby, right? So for those reasons, use pea plants. Now, how would you all test this theory that there is a blending of characteristics when you form offspring? What would you do with pea plants? Exactly, right? So what he did first was he spent years developing purebred lines. What does purebred mean? Just layperson terms. How would you describe it? Only one characteristic, right? Purebred organisms they've been manually bred for years to have a certain set of desirable traits. Okay, so what he did first was he spent years creating purebred lines, only tall and only short pea plants. Now remember, at this time they had no clue that DNA was the molecule that carried your traits. All they knew was that there were factors. Traits were coded in factors. Somewhere in a cell, these were these factors. And so, by doing these purebred lines, what he did was he created completely tall and completely short plants, which he was sure only held the tall factor and the short factor respectively. Does that make sense? Good. Now, let's go a little bit back to reproduction. How do you actually force a plant to reproduce? Tell me about cross-pollination. Abhishek, in terms of the anatomy, how do I cross-pollinate a plant?
It's something that has the stigma.
Yeah. Stigma, style, ovary. That's the female reproductive system. What's the male one? Good, right? Which has an anther. So I'll quickly draw it for you. So the flower of a plant is its sexual or reproductive organ. And at the center, you have the stigma, style, and the ovary, which has the ova right here. Lookman, what about the male part of a plant? What does that contain?
The
male
filament,
part?
the filament and anther.
Good. Okay. And let's go to Georgina. Tell us what happens between the anther and the stigma, style, and ovary.
So the anther is pollen,
Yeah.
the pollen, and then and the top of the sticky, and then it travels down to the egg and it becomes zygote.
Good. So you form a seed, and the seed has to then disperse to actually form another plant, right? So how could you all modify this process? Think about it. You're Mendel. You've got forceps and you've got a brush. What do you do? Exactly, right? So he got a brush, he took pollen from a male plant. Remember, plants are hermaphrodites, had male and female organs, right? So he got pollen from the male plant, and then the plant that he decided would be the female or the seed-carrying plant, he then fertilized it. Now, can plants self-pollinate? So how do you prevent that happening? Because once you've created these purebred lines, you want to mix the tall and short plants to see whether you get a blending or whether something crazy happens. You don't want the short plant to accidentally self-pollinate itself, because then you're just going to get more short plants. So how could you remove that confounding variable? Good. So that's what he used the forceps for, right? He removed the male part of the plant from the female plants, and he removed the female reproductive organs from the male plants. Good. This is important because you can get asked the HSC question about Mendel's techniques that he used, especially in the trials, much less common in the HSC. Okay? So those are a few things. I've written down all of this in his methodology, so not much to write. Okay? Do we understand what he's doing so far? Good. Now, what do you think he observed? Find the tall and short. Something crazy happened. Do you think plants are all tall, middle height? Mixed. Okay. Agree. Was there any ratio?
One would be more dominant and the other would be more recessive.
Okay. But remember, when do you get dominance and recessiveness? Go back to your understanding of alleles, right? A plant that's tall only has tall factors. So if we were to draw its alleles, it would be capital T, capital T. Plant that's short only have little t, which is a short allele. If they're combined, it will always be one tall with one short, right? So would you ever see a short plant? Think about it. Why is it called a recessive allele? It's because in a heterozygote where there is a dominant and a recessive allele, the recessive allele is completely masked. Does that make sense? So that is why this is your recessive allele. Now, what he observed was quite unique. All the offspring plants were tall, every single one. He did this thousands of times. That's why his work is universally known. His reliability was off the charts. He did this with thousands of pea plants. He's very bothered. So these are all tall plants. Can you explain why? Using your current understanding of genetics. Any ideas? We kind of just went through this.
Just if you were taking a plant and a short plant, and a tall is dominant most of the plants.
Good. Now, this is something I want you all to distinguish. There is a word called genotype and a word called phenotype. If you start blending the two, you stuff up biology. What is genotype and what is phenotype?
It's what allows is functional, and then phenotype is how it's expressed. So there's like environmental.
Good. Very good, but I'll dumb it down for us. Genotype equals allele combination. What set of two alleles do they have? It's that simple. Is it capital T, capital T? Is it capital T, little t? Is it two little t's? Phenotype starts with a P, so remember physical trait. What is the trait that came out of those two alleles? Right? And of course, as Pranav mentioned, genotype plus environment equals phenotype. Okay? Very good. So these plants are all heterozygotes. Do you all agree? They all got one capital T. I'll zoom in for all of us. We all got one capital T from the tall plants and one little t from the short. So all of them are heterozygotes, and the tall or capital T will dominate the recessive allele and thus be physically expressed. Does that make sense to everyone? Any questions thus far? Good. But just remind yourselves, Mendel had no clue that there were two factors for a trait. He had no clue about DNA. So imagine how confused this guy would have been, seeing all these plants were tall and not mixed as what biology said at the time. What would you do next? Sushila, you're a curious scientist. What should you do next? You're on the verge of developing the 3 laws of biology that will change the world. This is the groundwork for genetic engineering and CRISPR. So you're going to make it or break it. What's the next experiment you do? No? No idea? You guys are too scared. Go out there. Just make a bold, curious. There's no right or wrong. It's science. It's all about curiosity. Okay, so I'm going to start using names for this, right? So these are the parents. And this is the F1 generation, or first generation. So which plants are you talking about? So what are you going to do to them? Okay, you could breed them with a parent. Okay, Abhishek?
I'll go and breed them with each other.
Okay.
With each other.
See, the thing is, if you breed them with a parent, Mendel knew the parents only had the tall traits, and he had no clue what the offspring had. So his emergent first step, neither of you are wrong, but his immediate next step was to breed the offspring with each other. Because the goal here was, I want more info about the offspring. Why are they all tall? What genetic traits do they have? Right? So he decided, if I breed the offspring together, maybe I'll get some clues into what's going on. And this led to a very, very famous ratio. Can anyone guess what happened when he bred the offsprings together? He did this thousands of times as well. Just what happened? What would you observe?
I would say that since the F1 generation has the recessive allele as well, they would have at least one.
So if there's 4 plants, you're telling me one of them will be? How did you come to that conclusion again? Oh, he's mentioned a big word, Punnett Square. Who's done a Punnett Square school? Raise your hand. Okay. Who knows what a Punnett Square is for? When do you do a Punnett Square? Because another word that confuses students is pedigree. When do you do a pedigree? When do you do a Punnett Square? We'll come to those definitions, but for now, since you've said the word, we have to define it. Any idea what a Punnett Square is, Abhishek?
Are we doing it like
Okay, so good memory, right?
But I think it's just like a box of 4, and you have the alleles. You have like dominant on the
Can you tell me why it looks like that? So I'll tell you all what he's saying, right? So you'll agree that genotype, going to our current understanding, the genotype of all these plants is heterozygous, capital T, little t, right? So if you were to do a Punnett square, it looks something like this, where you draw both the parent genotypes, and you literally look to the left, whatever letter you see, you just draw it out like this for you. Whatever letter you see on the left, write it down. Whatever letter you see right on top, you write it down. It's a mindless task. like this. And as Pranav said, we notice that one of the organisms, one of the offspring, but out of every 4 organisms, 1, 2, 3, 4, one of them will have 2 little t's. Is there a dominant allele in this genotype at all? So what's going to happen to the recessive allele? It's not being masked anymore. It's going to be the phenotype. Remember, this is genotypes. What will the phenotype be? Short. Okay. Now my question to you is, why do we do it like this? Why do we do a table and we look up, we look to the left? Give you all a hint. This is modeling a process that you all studied in a lot of detail. Yeah, do you all see? This is literally a very simplified model of meiosis. What happens in meiosis? Homologous chromosomes move to opposite ends, and so do their alleles. And eventually, each gamete randomly inherits one allele. So this gamete randomly inherits one allele from one parent, the other allele from the other parent. Right? And we do it 4 times, so we look at all the possibilities, because there's only 4 possibilities that can occur. Does that make sense? So what Punnett squares are the very simplified models of meiosis. They model random segregation. Do you all see that? Do they model independent assortment? Why do you say that, Abhishek?
Yeah, they don't model independent assortment because that's what the separation of the president is just a alignment problem.
Wouldn't you kind of assume that there is already
Ron, what do you think? Are we modeling independent assortment or not? This would be a good HSC question.
I don't think so, sir. I think it's more so random segregation because there's only one. It's like we're only looking at one trait, or like one chromosome.
Good, that's what I wanted to hear. Did you all hear that? What is independent assortment? Independent assortment says that the alleles of one gene, or the one homologous pair of chromosomes, randomly segregate independent to another homologous pair, which has a different gene. Different genes are different traits. We're only looking at height here. Do you all agree? So this is solely modeling random segregation in meiosis. Good. So now do you understand why a Punnett square looks as though it does? Good. So now my question is, when do you do a Punnett square? In what scenario? Good. So when you know the inheritance pattern of a trait and you're asked for offspring genotypes and phenotypes. So if I had to dumb this down to one word, you do a Punnett square to determine offspring genotypes and phenotypes. If a question asked you, what is the inheritance pattern of this disease? Do you do a Punnett square? Did it ask you anything about offspring in that question? No, so no Punnett square. Does that make sense? Bit of an extension of the question. What do you do then? They ask about inheritance pattern of a disease. Yeah, so pedigree is kind of the step before a Punnett square. See, in this scenario, I already told you all what the tall or the dominant allele or the recessive allele was. You knew the inheritance pattern of height. Tall is dominant, short is recessive. But when you don't know that, you need to do a pedigree first. So the goal of a pedigree is to determine inheritance pattern. The goal of the Punnett square is to determine offspring genotypes and phenotypes. Does that make sense? So if you're a scientist, you discover disease X, the first thing you do is a pedigree. What's the dominant allele? Is it the diseased or is it the normal? And once you determine that, you want to know if person A and person B have a child, what's the likelihood the child will be affected? Then you do a Punnett square. Do you guys see the progression there? That's what you do in medicine and genetics. Okay. Good. We'll come back to all of this. I'm just going a bit extended. But you must know this ratio. This 3-to-1 ratio. Now, this is what we call a monohybrid cross. Does anyone know what a monohybrid cross is? What does mono mean? What does hybrid mean? Mix. What are these organisms? Or these plants? Aren't they a mix of 2 alleles? Monohybrid cross. And it's a cross because it's a Punnett Square. Cross is a Punnett Square. So that's what a monohybrid cross is. It's a cross of two heterozygotes. Does that make sense? Good.
Excuse me, sir.
Yep.
So, what is the significance of that ratio? Like, why is that important to us?
Very good, because I'm going to ask you now. See, this is the thing you need to all make sure you try to do. All of this stuff that you're doing right now, Punnett squares, alleles, and genes, you're cheating. You're using knowledge of 2021 to solve an 1800s problem. Think about what Mendel thought. He had no clue that there were two factors. He had no clue about independent assortment and random segregation. They didn't even know the nucleus carried genetic information, that they thought it was proteins in the cytoplasm. These guys were clueless. So from that, I want you to kind of step back into Mendel's shoes, where you know nothing, right? I've taken your intellectual weapons of meiosis, independent assortment, and random segregation away from you. You know nothing. Think about it. Think about your economics friend who knows nothing about biology. If we gave him this experiment, what conclusion could he or she come to? This is why it's so hard.
Obviously, like the guy, Mendel did it a lot of times, right?
Thousands of times.
So each time he did it, he would have like seen that.
This is his results. Look at it. You can see he did it with many different traits. He didn't just do it with plant height. He did it with flower position, pod shape, seed shape, flower color, and he always got an F2 ratio 3:1. What does that mean? God likes 3:1s. What does that mean, 3:1? Why did he? What conclusion would you come to if you saw this? And remember, you can't use alleles, you can't use chromosomes, you can't do any of that. And this is the beauty, and this is why you're learning about Mendel too, like hundreds of years after his experiments. Do you all agree this is knowledge, right? Learning this is all knowledge. But there's a saying, knowledge is power. But I want you all to modify this. It's not knowledge is power, it's organized, applied knowledge is power. You guys all have this knowledge, the same knowledge that Mendel did, but your goal now is to organize it into something that you can actually have implications for. And the person that does that is the person that has power. So let's see who can come to a conclusion from this. Lookman, any conclusions you came to? Because you asked the question.
That the but you said not to refer to alleles, right?
Yes, and I'm making it very hard for you guys. Mendel didn't just look at this and go, I know the answer. It took him weeks. So I'm giving you guys 5 minutes
Okay.
to do it.
The conclusion would be that both parents have the, I don't know.
Yeah, see, it is hard. That's what I want you to understand. I don't want you to come to the conclusions he did, because it's virtually impossible to. Mendel discovered the only way to explain this was using 3 laws that he developed. Okay? I'm going to introduce you to those 3 laws. These 3 laws explain these observations. These are his 3 laws. The first law is a law of dominance. It's something that you've already been introduced to. Now, Mendel called alleles. Remember, he had no clue about chromosomes, about DNA, all of that. The craziest thing was decades later they discovered chromosomes, alleles, and they went, what Mendel was talking about was the allele, and he knew that hundreds of years before we even discovered it. He called alleles genetic factors. Genetic factors. Okay? Because he had no clue what they were. And his conclusion was that every organism has 2 genetic factors. And there will always be one genetic factor for a trait that is dominant over another. That is a law of dominance. Does that make sense? That's what you all learned. In a heterozygote, one allele will be dominant over another. That's the law of dominance. Okay? Very good. So right, each parent carries 2 genetic factors, all alleles. This is kind of like the first sentence for the law of dominance. Each parent carries 2 alleles, one allele would be dominant. But remember again, he had no clue what alleles were. I want you to pause for a moment. What does a law mean? Because there's theories, there's laws. You learn about the theory of evolution, but you learn about the law of gravity, and physics has a lot of laws, right? So what's the difference between a law and a theory? A law is universal and proven, right? A theory is a potential explanation for observations. Why is evolution not a law? Why is natural selection not a law? Yeah, but there are laws that are not universally accepted. Earth is round is pretty much law, but people still believe in flat Earth. Doesn't mean we call it theory with them. Yeah? Think about this. How long does it take evolution to occur? And how long does a human live? Hundreds pushing it. The average life expectancy is 84 in a developed country, right? So no human has lived long enough to actually see through evolution. So we're just using it based on historical data, etc. That's why evolution is still a theory. Okay, good. So that's the law of dominance. Now, this is the thing, law of random segregation. I want you to just realize, was it very easy or was it a little bit challenging to understand random segregation in independent assortment? Yeah, and that's when you had a picture of chromosomes, you had an entire cell with a nucleus. Now imagine if all that got wiped out. How much harder that would be. And this guy defined or explained random segregation without ever seeing a cell in his life. He's not even a scientist, he's a monk. He didn't study science. Okay? He already, years before we looked at the movement of chromosomes, hypothesized random segregation. He said, during the process of reproduction or meiosis, or cell division of those cells, those two genetic factors for each individual will separate. That's what random segregation is, right? Because the two factors or alleles sit on homologous chromosomes. And what do we say about chromosomes in a homologous pair? They randomly move to the opposite end of the cell. That's what the theory of random segregation, or the law of random segregation is, which each allele, or what he called a factor, randomly segregates in an organism at some point in time. We know now it's an anaphase 1 of meiosis. Very good. Any questions so far?
Yeah, excuse me, sir.
Yep.
What was the conclusion again with the 3-to-1 ratio? It's that.
These three laws were his conclusion.
Okay, but previously you said that each parent carries two genetic factors which one that dominant, right?
Yeah, that's right.
So what about the parents who carry homozygous?
Yeah, so what we said was that each parent carries two genetic factors, and we said that one factor will be dominant over the other. We didn't say that every parent must carry a dominant and recessive factor, or dominant and recessive allele. In the case in which they do, one factor or allele will be dominant, right? So just because he mentioned the law of dominance doesn't mean every organism must have a dominant and recessive allele. It just means in the case that an organism has this genotype, the dominant allele will overexpress over the recessive allele, right? He doesn't dismiss that you can also have this genotype and this genotype. And what would you call these two? This is heterozygous. What do you call the other two? Homozygous dominant and so all he mentioned was that in organisms which have a dominant and recessive allele, the dominant allele will be physically expressed and mask the recessive. Does that make sense, Lachlan?
Yep, thank you, sir.
Okay, good. Right. Now, we've explained the law of random segregation, and finally, we also explained the law of independent assortment, which is what you already know, what happens to chromosomes of different homologous pairs. It's just, he concluded that from this. What he realized was that whether the plants were tall or short did not affect the inheritance of seed shape. Whether the plants had a different pod color had no effect on the inheritance of flower color. And so he logically came to the conclusion each trait is inherited independently. And we now know that because each trait or each gene usually lies on a different homologous pair. Does that make sense to all of us? Good. So that was his third law. So if I were to draw it out for you, what Mendel's saying for the law of dominance is that if an organism had a heterozygous genotype, the dominant allele would mask the recessive allele and be physically expressed. What he's saying in random segregation is that whatever the genotype the organism has, each factor or allele will separate. That makes sense. And the final thing he's saying is that trait T is inherited independent to trait R, which could be, let's say, plant height is trait T, and let's say pod color is the whole different trait. Good. Now I'm going to give you all a minute. I want you to very quickly write one line to explain each law in terms of meiosis. So what is a correlation in meiosis? So don't worry about the law of dominance. Explain random segregation and independent assortment. How does the statement he's made make sense in relation to the movement of chromosomes? And I will ask Raya and I will ask Harshil in a minute. Be ready with your answers. I don't want you to regurgitate the definition of random segregation and independent assortment. I want you to explain how it makes sense with reference to the movement of chromosomes. Pretty much what we've already done. Anyone have any questions at all so far? Makes sense. Has anyone already learnt all of this at school? Okay, good. Module 5 is a huge module. Usually we finish a module in a term, but Module 5 always will spill over to the next year because it's humongous. You'll notice Module 6 is much shorter. Okay. Okay. Let's start. Yes.
So this is not about the question, but isn't module like 7 and 8 the biggest modules, or is it?
I would say Module 8 is much bigger than 7. 7 is not too big. It's a bit challenging because you learn about the immune response, which is very interesting. I think Module 7 is probably the coolest module. Module 8 is very big. It's kind of got a very big bottom end in the sense that you learn about the eye, the ear, and the kidney. You have to learn what's normal, what's abnormal, and technologies to treat it. So you become a specialist of those organs. And that takes ages for students, especially. And the other thing is, they've never asked a question in the HSC about the eye, the ear, and the kidney. And it's a huge part of the syllabus. So very likely, your exam will have a question about technologies and disorders. I was anticipating it was last of this year, but this year was just apparently a breeze for an exam. All right, let's start going through this. So, Brad, what's your explanation for random segregation? Why is it that Mendel mentioned that alleles for a trait randomly separate or segregate in an organism? What's your answer? Quickly. Okay, we'll move on. Harshil, what's your answer for either random segregation or independent assortment? Okay, either we're shy or we're not, mic's not working, but make sure you both have your mic working. Okay, I don't want to ask you all questions and get radio silence. Let's go to Georgina. Do you want to give it a go?
I was trying, but I was just like,
That's all right. Okay. Let me ask Varun. Give it a go. Random segregation. What would your answer be?
So random segregation. So the random movement of chromosomes. It increased like genetic variation through.
Pause. You don't want to explain why it's increasing genetic variation. You want to explain exactly why Mendel mentioned genetic factors will separate in an organism. To link it to those alleles. Where do those alleles lie? No? Give it a go.
And when during phase 1 when they move to opposite ends of cell, alleles also move to opposite cell, which is
and that does randomly segregated to gametes. Good, very good. The only thing I would say is alleles of a gene lie within the homologous pair. Just within would be the word I would use. Good, so my answer would be Yeah, these genetic factors or alleles lie within a homologous pair of chromosomes. In anaphase 1, the homologous chromosomes segregate to opposite ends, and thus so do these genetic factors / alleles. And I know I've explained it because I've used thus, right? If you use thus as a result of due to, you've explained it. Okay? Good. Who wants to explain independent assortment? Sashira, give it a go. How would you explain independent assortment? All right, just give it a go.
Since the movement, the positioning of one of the doesn't affect the other.
Good. So that's independent assortment. How can we link it back to these alleles or genetic factors? Because that's what you need to be doing. Because he's talking about genetic factors, you're talking about chromosomes, you need to unite the two. That's when you explain things, right? So, yes, Tashbia?
Independent of each other, so the alleles. So then the gene, the alleles are co-specific gene will also be separate.
Good. Yeah, I think a key thing I'd start off is saying that alleles for one gene lie on a different homologous pair of chromosomes to the alleles of another gene. Thus, when the chromosomes of two different homologous pairs independently assort, so with the alleles for those two different genes, aka traits are inherited independently. Yeah. Yes, see, this is a very good. See, I like when you all think critically. And I'm not here to spoon feed you, I'm here for you to make your own conclusions. So, Pranav is completely right. There could be a gene for height here, and the gene for eye color could be right here. If that was the case, what would you observe? Would those two traits be inherited independent, or would they be always inherited together? Let's say this person is tall and they have brown eyes, right? Are those traits now going to be inherited together? 100%.
Wouldn't it depend on where it gets cut?
Yes, and that's the exception, right? So it will always be inherited together unless something in prophase 1 of meiosis happens right exactly at that point and breaks them off. Very, very low likelihood, but it can happen. Okay, good. So these are all thought experiments that you need to understand, which is if two genes or two traits lie on the same chromosome, always inherited together, as crossing over would dissociate them. Okay, very good. Yeah, that's pretty much everything you need to understand. But Pranav, statistically speaking, a human has how many chromosomes? 23 pairs of 2. What is the likelihood two genes we're talking about lie on the exact same chromosome? It's much more likely they lie on one of the other 22 pairs. Statistically speaking, it won't be the case, but there will be some cases where two genes lie on the same chromosome. Okay? Now, last thought experiment for all of you. How would it change when we're looking at a gene that lies here and a gene that lies here? Let's say this is, I don't know, what's a trait humans? Skin color. Let's call that skin color. How will the inheritance of height vary or be aligned with now hair and skin color? What's our answer for that? Think about this, they're on the same chromosomes, they're always going to be inherited together, but the exception is if crossing over happens, right? Now, which two genes are more likely to be dissociated? T and R or T and B? T and R, because crossing over can happen here. It'll all separate T and R. But only a breakage right there would separate T. The likelihood of crossing over dissociating T from R is much, much higher. Does that make sense to all of us? Good. So if we look at this statistically, we see that height is inherited with skin color 80% of the time, and 20% of the time it's not. Whereas with height and hair color, we see 99% of that time they're inherited together, and 1% of the time crossing over happens exactly between this bar and this bar. Okay, good. So these are all critical thinking thought experiments you should all have, because you can get HSC questions about this. Very good. All right, let's move on. Let's look at, okay, a few words I just want to introduce here. Now, this is called karyotype analysis. Has anyone heard of karyotype analysis before? Never. Now, what you do in karyotype analysis is you get a cell, you look at it under a microscope, you wait till all the chromosomes have aligned in that cell. So what phase would you wait for, Tanisha?
Wait, repeat it.
All the chromosomes have aligned in a cell. What phase? Good, right? So wait for metaphase. Now, what do we do at metaphase? We freeze the cell, we kill it at metaphase, and we take a photo of all the chromosomes. And kind of like how you did in kindergarten, you cut out all the pictures and put it in size order. That's literally what this is, right? So this is just a picture of all the chromosomes. It's radiolabeled as well. That's how they take the picture. They use radioisotopes. But then they cut out the image of every homologous pair and they align it with each other. And you see, it's always, chromosome pair 1 is always the biggest pair, and it goes smaller, smaller, and smaller. But look at there's one weird pair you notice if you look at these karyotypes. They don't have numbers, they have X and Y. These are what we call your sex chromosomes. These are what we call, the rest of this entire genome is what we call your autosomes. Autosomes are body chromosomes. Now, everyone, the only reason we're making this distinction is because sex chromosomes, the reason they're called sex chromosomes is because they determine gender, right? They determine if you're male or you're female. And because they're unique and different to your body chromosomes, right? If I told you all that the allele for eyebrow thickness was here, where would the other allele for eyebrow thickness be? Right here. Does everyone agree? But the issue with this X and Y is, do you see how the Y chromosome kind of looks like a degenerate, broken down version of the X? Half its size, and it doesn't have many alleles at all. In fact, whatever alleles are on the X chromosome, none on the Y. The Y has a very specific set of about 6,000 genes that only code for male development. Okay? So if someone was XY, what would their gender be, or their phenotype? Male. What about XX? What about X? Real hard question. That's a disease. It's called Turner syndrome, where it's a female because they don't have a Y, but they have very impaired reproductive function. Their ovaries fail when they're a teenage age. They become very short. They have a webbed neck and all these weird features. What about XYY? What would you expect? A masculine male. Yeah. So there are people you may know who are XYY and never know, because they'll actually be normal, except they'll naturally have more muscle mass or secondary male characteristics, especially after puberty. Yes?
What about people who are like intersex?
So there's many different causes for being intersex. So it can be if a male is XXY. So what would you expect to see in a male who's XXY? More female characteristics in a male. So a male that can develop breast tissue, a male that has abnormally smaller reproductive genitalia, XYZ. It's called Klinefelter's syndrome, right? So just so you understand the importance of the X, the importance of the Y chromosome, I'm giving you all extremes. Intersex is a very complex topic. You would all know someone who's intersex, because every 1 in 100 babies are intersex. And think about how big your school is. There would be intersex people, statistically speaking. Now, that's not just due to genetics, it's because of hormone deficiencies that can happen. So there is, I don't want to go into the detail, but on top of the kidneys, there is a structure called the adrenal glands. Has anyone heard of those? They release hormones. When things go wrong there, intersex can occur. Okay? Good, but that's way out of the syllabus. What would this person's gender be? Very good. So for Y, that's simple. Okay, good. Now, I guess an interesting point for all of you is all of us in utero, in the uterus, we're all female by default. All the males, all female. And what happens is, at a certain point during development, the Y chromosome will activate and start producing protein via transcription and it makes hormones, and those hormones will change those female reproductive structures into male reproductive structures. In fact, if you look at the female reproductive system, the anatomy, very similar to the males, except it's almost inverted. If you think about this, right? So that's literally what those hormones do. The interesting point is everyone is female in utero, it's just that hormone that changes the entire physical trait. Very interesting. All right, let's move on. Let's talk about inheritance patterns. So autosomal recessive, autosomal dominant. What did we say autosomal meant? Are we talking about the sex chromosomes or the autosomes? Exactly. So that tells me this gene or trait lies on one of the first 22 pairs of chromosomes. That's what the word autosomal means. What does the word recessive mean? The trait we're talking about is coded for by the recessive allele. So if I say cystic fibrosis, which is the most common genetic disorder a human can have, 3% of children have a genetic disorder, and the most common out of that is cystic fibrosis. That follows this inheritance pattern. What does that mean? It means the gene lies on the first 22 pairs, and it means it's coded for by the capital letter or the little letter? Little r. So what this means to me is a little r equals disease. Okay? And it lies on a body chromosome. Do not use the word body chromosome, the word autosome is more common. Good. So, Varun, what would it mean if we're talking about an autosomal dominant condition? Break it down the same way we did.
Yep, so the dominant allele is what it determines the if it's like
Dominant is disease. That's what it means, right?
Yeah.
So if it's autosomal dominant, let's start with the first word. It's an autosomal inheritance, so it lies in the first
1 to 22.
Yeah, and it's dominant, which means capital R equals disease. So the dominant allele is a diseased allele. An example of this, diabetes is very complex, and it's not just one gene. So it's thousands of genes interplaying with the environment. But I'll give you a very simple example. Who's had a Huntington's disease? Does anyone watch medical shows, medical TV dramas? Yeah, which one do you watch? Okay, I don't watch any now because once you actually get into it, you realize how medically inaccurate it is and it gives you a migraine. But coming back to this, there was an episode in House. I think you're also young, but have you heard of House, MD? It was a really good show, but it explored Huntington's. And basically, Huntington's is a disease where by the age of 60, you start getting symptoms, and it's 100% likelihood of death. And what happens is you get abnormal jerky movements called chorea. They get neurological impairment. Their brain cells start to die. Okay? And it's a dominant pattern of inheritance. So what that means is even one of your parents had it, you're very likely to get Huntington's. Right? So you will live your whole life knowing at about age 55, you'll start to get these neurological symptoms and die shortly after. So it's pretty much a death sentence that you know from birth. So that's the life impact of this autosomal dominant conditions. Good. We spoke at Punnett Squares. Let's move on. And let's talk about non-Mendelian inheritance. Now, out of these, the most important that will come up in your exams is this one. The other two rarely come up. If they do, it'll be in trials. They're very easy as well. The most important one I want you to understand and focus on is sex linkage. Okay? Now, I'll give you a break after I explain non-Mendelian inheritance, but Tashkir, could you explain the ratios we observed? Think about this pea plant, right? So we had a tall plant and a short plant. In the first generation, what did we observe? Tall. In the second generation, what did we observe? Remember, you always have to say the ratio, which is 3-to-1 ratio. Now, if there is ever a trait in which we do this exact same experiment, where we get trait 1, trait 2, we combine them, and in the second generation we don't get a 3-to-1 ratio, we immediately classify this as non-Mendelian inheritance, because it does not fall under Mendel's 3-to-1 ratio and cannot be explained by his laws. Okay? These are the 3 examples of non-Mendelian inheritance. The most important being sex linkage. Now, does anyone know what sex linkage means? What were the sex chromosomes again? They were the last pair, the XX and XY. And what did I tell you all? Out of the X and Y, which one of those two, for the HSC exam, carries the alleles? The X. So this is a useful one. This is a useless one, only for male development. Okay? So if a gene or a trait is sex-linked, what does that mean? The gene lies on X chromosome. Now, normally, for one of Mendel's genes, how many alleles does an organism have, everyone? So if I'm talking about height, how many alleles do those pea plants have? Each pea plant had 2. But think about this now. Imagine that height lied on the X chromosome. How many alleles would a female have who's XX? 2. How many alleles would a male have who's XY? 1. You see how it stuffs up everything? You can't even do a proper Punnett Square anymore. So that's why you deviate from the 3-to-1 ratio. It's because females will always have, I'll write this down now, but it's also in your notes. Females always have 2 alleles. Males have 1 allele. Can males ever be heterozygous for an X-linked condition? X-linked or sex-linked is the same thing. No, they can't, because they only have 1 allele, right? So do you see how there's a lot of implications from this? Good. On that note, hit the hour mark. I want you to stand up, stretch. Feel free to go outside, get some fresh air. We will resume at 12:16. Okay? If anyone has any questions, please do ask.
Excuse me, sir.
Yeah.
Um,
Give me 1 minute and I'll answer your question. Just 1 second. Okay. What was your question?
So why do males have 1 allele? Is it because of the Y chromosome?
Because the Y chromosome is a very degenerate or broken down version of the X, and it does not have many of the alleles that the X chromosome does.
So doesn't that mean the males will always inherit the disease?
Yes, and that's what you'll see. So with X-linked conditions, you'll see males will be much more affected. So if you ever look at, have you done pedigrees before?
Yeah.
So you'll see in those pedigrees, it's only males who will be affected. If you ever see that pattern, you should be thinking, is this a sex-linked disease?
Okay.
Good implication. All right, we will resume at 12:16. All right, let's jump back into this. So we're going to talk about the other laws before we talk about sex linkage, because they're quite easy and straightforward. Now, we'll begin with codominance. The word, if you think hard enough about it, explains this pattern of inheritance. In codominance, everyone, there are 2 alleles that are equally dominant to one another. So let's imagine we're talking about cow hide color. This is a very famous example of a codominant trait. You can see one of the alleles is capital R, right? And they've written C with R as a subscript. Can you all see that? C for color and R for red. You can also see they've done C for color and W. Now, W is a little bit hard. You can't tell if it's lowercase or uppercase, but I'm telling you all this is uppercase W, right? And this is to show you that there are 2 equally dominant alleles for this gene. 2 different letters, that's the way you do it. Now, what do you see in the offspring cow? Do you see a blending of colors, or do you see 2 dominant alleles aggressively fighting it out and winning a battle in different parts of the cow? That's exactly what we see. There are patches of completely red dominant hide, and there are patches of completely white dominant hide. So you need to remember this. With a codominant trait, there is no blending at all. It is just patches of one allele fully being expressed and patches of another allele being fully expressed. So my question to you is, how many phenotypes can you get with a normal pea plant gene? Let's talk about pea plant height. What are the 2 phenotypes you can get? Tall or short? It's a binary. Do you all see that? With a codominant trait, how many different phenotypes can you get? So this is the thing. This is one phenotype. It doesn't matter how much white or red it is. If they have patches of white and red, they're what we call the Rowan cow. This is a very famous biology example of a codominant trait. So you can be white, you can be red, or you can be Rowan in cattle color. So there are 3 phenotypes. Does that make sense? So I want you to write this. There are going to be 3 phenotypes. And I want you to also write this as well. There is no blending at all. Characteristics. No blending. There are patches of allele that fully express. Now, a very high yield example for the real world of codominance is the ABO blood system. Now, is anyone interested in medicine here? I know Pranav, you mentioned. Kashvia, would you ever consider medicine? What's your reason?
I guess it's just nursing just feels much more to me.
That's good. I think nursing is an amazing career as well. Either way, you could be a nurse practitioner and you get to do the specialist work. That's really cool. Yeah. Coming back to this, the reason I brought this up was if you want to do transplant surgery or hematology or literally any medical specialty, you will have to have a good understanding of ABO compatibility. Right? Imagine you're working in the emergency department. A patient comes in with massive blood loss. That happens at Liverpool Hospital, where there's actual gunshots and all that happening, right? In that case, you need to make sure you give them blood products to save their life. But if you give them the wrong blood type, you will kill the person, right? So it's very important to understand how ABO blood system works. Now, the ABO blood system is not just codominant, it is even more complicated in that there are 3 alleles for a gene, okay? I want you to go back to codominance. How many alleles are there for a gene? 2, right? There's red or white, and when they occur together, they make a different phenotype. But there's only still 2 alleles. With the ABO system, there are 3 alleles, and 2 of them are codominant to another, which is recessive. So what that means, let's draw it out and understand this. We're talking about red blood cells here. You might have remembered from year 7, red blood cells are a biconcave disc, right? Where they're a bit shallow at the center. And if you look at it bird's eye view, you'll see something like this. Now, if you zoom into the red blood cell, it has marker molecules, right? Kind of like in kindergarten or year 3 where you all had pencil cases with your name on it, where you can just slot in letters which tell you, it's your pencil. It's ownership. These are particles that convey ownership, that this is yours, this is self. In the same way, all your red blood cells have what we call marker molecules, right? The name of these marker molecules, you might have heard them before, are called antigens. Antigens are just marker molecules. Every one of your cells is coded with your antigens. That tells your body, this is mine. And everything outside of that, whether it be dirt which falls into a wound in your arm and you get hurt, all of that is foreign. Do you all agree? Even the COVID virus, when it enters the body, your body recognizes it as foreign. What does the body do to foreign objects? It attacks them. It tries to destroy them. So it's so important that your body has self-antigens or self-marker molecules, and it does not attack them, and it attacks foreign marker molecules. Does that make sense? Very good. So that's what these antigens are. Now, there are 3 different variations which can occur. We can have A antigens. We can have B antigens. Remember, these are just sugar molecules. If you zoomed into the chemistry of this, these are carbohydrates, kind of like the sugar in DNA. That's what these antigens are. And finally, you could have O is no antigen. That's what O allele codes for. No marker molecule at all. Okay? So those are the three alleles you can have. Have the A allele, which codes for the A antigen. The B allele, which codes for the B antigen. And the O allele, which codes for no antigen. Good. Now, the only second thing you need to understand is that the A and the B alleles are codominant. Okay? Let's talk about what this all means. So let's have a look at how we denote this. Now, everyone, when there are more than two alleles for a particular trait, we have to start subscripting things. Do you all see how we're subscripting I and we're putting an A, a B, or an O there? That's because we can't just be writing A, we can't just be writing B and O, because everything looks dominant here. Do you all see that? So to simplify and understand this better, we will subscript it whenever there's more than two alleles. Okay? So let's start with this genotype. Let's understand this, right? Imagine you're working in the emergency department, you're a junior doctor, it's your first day, someone comes in, they're dying of massive blood loss, you run a test and you find that they are this particular genotype. What would their red blood cells look like? Let's draw this. If we had their red blood cell, this is the biconcave disc of the red blood cell, what antigen would exist on its surface? AN? Good, only A antigen, very good. Only the A antigen, because there's no B allele. Okay? So this person's red blood cell is peppered full of this A antigen. Okay? This is all their body knows, and this is what their body identifies as self. Now, if I were to give this person blood with B antigen in it, would their body recognize that as self or foreign? And what would the body do to anything that's foreign? And in that process, their blood would clot, you form a blood clot, and that can cause a heart attack or a stroke and kill the person. Okay? So my question to you is, what kind of blood would you give them? Blood with what antigen? Yeah, you could give them A, very good. So logically, their body recognizes A as self. Or, very good, you could give them red blood cells that have no antigen, which is O type blood. Does that make sense? So that's how we understand this. So remember, we need to separate genotype and phenotype. So the genotype is AA, but their phenotype is blood type A, which means their blood only has A antigen. Who can they donate blood to? That's my question now. What phenotypes? Other A's and any others? Very good. Remember, if someone was AB, let's go back to very quickly have a look at someone who's AB. So imagine that they are IA, IB. Their red blood cells would contain what two antigens, Ashita? Yeah, right. So there'll be some A antigens, there'll be some B antigens. Remember, that's what codominance is. There's no mixing, just patches of one antigen, patches of another, right? So their body recognizes A and B as self. Everything else there's nothing else, right? There's O, which is no antigen. So these people can receive A positive blood, they can receive A blood, B blood, or they can receive O blood, which has no antigen. It doesn't matter for them, right? So if you were to lose blood or get in an accident, being AB is the best blood type you can have because you can take blood from everyone. Can you give blood to everyone? You give blood to an A person because you have B antigens, which are going to trigger an immune response. Can you give blood to a B person? Can you give blood to an O person? They've never seen an antigen in their life. You give them A and B, they destroy their body, right? So it's kind of genetically selfish trait to have. It's evolutionarily favorable, I guess, because you need blood from everyone, but you can't donate to anyone except ABs. Very good. Yes?
How do people get AB blood types?
We'll talk about that. It's to do with the inheritance. But let's quickly go through the other blood types very shortly. So you're completely right. So don't worry about antibodies for now. Antibodies are just proteins that we make to destroy foreign things. So as we said, this person is an A blood type. If we give them B blood, they would generate antibodies to fight those B antigens. That's it. So it's all an antibodies. So we'll skip this entire section for now. Yeah, hopefully you all got that they can donate to A and AB. That's what I spoke about AB. AB has A antigens, which are self or non-foreign. Okay, good. What about if you're this genotype? What would your phenotype be? Good, because degree it's your simple dominance relationship. The A will mask the O, and the phenotype will still be A. It'll be the exact same effect as our previous example. Now, what about if you were, I'm sure you all know this, who can this person donate to? Good. And ABs, they can donate to ABs. Who can they receive blood from? Very good, good job. And remember, when you're this genotype, your phenotype will be the exact same because you'll mask the O. Very good, good job. So that's blood type in a nutshell. That's what you need to understand. Now, the word positive and negative, we're not going to talk about that right now. We'll maybe leave that for a little bit, but that's a whole different antigen system, which is inherited independently. So you can be A positive or A negative. They're not related in any sense. That's called a rhesus antigen system. We'll talk about that later. Okay, good. Let's move on to, you can do these questions in your own time over the holidays as well. And let's talk about sex linkage now. Any questions thus far, especially virtual students? Any questions at all? Gori, do you have any questions thus far?
No, not really.
Okay, good, good. And Georgina, any questions at all?
No, I'm okay.
Good. All right. Now, this is where I want you all to focus. This is very important. I'd say this is probably one of the most exam-high parts of this module, with understanding sex linkage. Okay? The key things you have to explain is why this does not follow a Mendelian ratio, and then how to do Punnett squares and pedigrees with it. So those are the three headings you should have in your head. Okay? Why is it non-Mendelian? How do I do Punnett squares with this? How do I do pedigrees with this? Okay? So we'll start with the first principle. Why is it non-Mendelian? Now, what did I say? Where does sex-linked genes, where do they lie? So, X and Y? Only on the X. So remember, the key feature here, if anyone mentions sex-linked or X-linked gene, that lies only, especially for the HSC, only on the X chromosome. Very important you understand that. Okay? Good. Now, some examples of traits that are sex-linked. The most common one is colorblindness, right? So you'll notice that the colorblindness lies in the X chromosome. So males are much higher risk at getting colorblindness, and we'll explain how that is based on X-linked inheritance shortly. Another more interesting condition is hemophilia. Has anyone heard of hemophilia before? Any ideas what it is? Anyone follow the royal family? Interested in Kate?
The blood doesn't clot.
Yeah, the blood does not clot. So what happens to these people is randomly they'll get deep muscle bleeding deep in the eye socket, and it can result in death if bad enough, right? So that's hemophilia for you. It's because they have a gene mutation which doesn't make a blood clotting protein. It's called factor VIII. It's one of the most important blood clotting proteins you need. So these people have a gene mutation that can't make that factor. And as a result, the blood is kind of in an anti-clotting state all the time. So it's prone to bleeding randomly in joints, muscle, in the eye, etc. Maybe even in the brain it can happen. Okay? Now, the reason that happened in the royal family, can anyone guess why?
Yeah.
Exactly, right? So a lot of marriage between biologically related people. Can you tell me why that's bad? You've learned enough genetics. Why is it bad to marry a sibling or a cousin or someone related to you, genetically?
They can have similar traits to you. So if you have a favorable trait, it can become dominant
Very close, very close. Not necessarily that will become dominant, but you all need to understand you have many thousands of genes, right? Now, all of you, including me, will have about 6 genes for which we have a recessive allele that could kill you right now, right? But right now it's being masked by a dominant allele that's protecting you, and you're not showing any symptoms at all. Now, every single person has a different set of 6 recessive alleles that are deadly. But if you marry or you have a child with someone related to you, it's very likely you both have the same 6 deadly alleles. And if you have a child and that child gets one recessive allele from you that's deadly, one recessive allele from the other person, they'll express the disease. In which case, one example is Down syndrome is completely different because that's a chromosomal disorder, but this is hemophilia would be one example of that. Down syndrome is not even due to incest. Down syndrome can happen to very normal people. Does anyone know what the main risk factor for Down syndrome is? Think reproduction, think ova, think females.
There's one extra.
That's what it is, but what's the main risk factor? It's going to impact your life decisions. Age. Of who? Mom and dad. Women are born with all their ova. So if you go, as we've spoke about born diabetes, 20,000 mutations per hour, the ova also experience those mutations, right? So what happens is a woman that's 40 has way more mutations in her ova, and the ova are broken down to the point where the risk of improper meiosis is so high, right? So you realize when a woman becomes about 45 years old, she won't have a child. The risk of Down syndrome is like 1 in 16. And the second you pass age 30, the risk of having a child with Down syndrome increases beyond 1%. So less than 30, it's less than 1% risk. The second you pass 30, it's not 1%, 1 in 1,000. The risk goes above 1 in 1,000. Yeah, so that's an interesting point. But anyway, we'll come back to this. So hemophilia is one key example. I want you to just write next to hemophilia. Mutation of factor 8. Factor 8, which is a blood clotting protein. Good. And we've already spoken about the fact that females are XX, which means they can carry 2 alleles for a sex-linked condition, whereas males are XY, which means they only carry 1 allele. That very fact makes sex linkage fall outside your 3-to-1 Mendelian ratio, and we'll talk about that now. Good. All right. So the next thing I want you all to understand, this is just generally with biology. Most diseases are coded for by a recessive allele, and it's the same principle when we are talking about sex-linked conditions. 99% of sex-linked conditions are what we call sex-linked recessive. Okay. So what that means is little r equals to disease. So let's apply a quick thought experiment, right? If a female were to inherit a little r, I'm going to put a little r here, does she have disease? It depends on what this allele is. If that allele is a capital R, everyone, what's going to happen? The dominant capital R will mask the disease allele, and the female will be completely normal. Whereas if in the rare case she inherits 2 recessive alleles, she will express the condition. Whereas males, they get a recessive allele, what does that mean? Game over, right? They have disease. So women have 2 rounds of this protective lottery. Even if they get a bad draw in the first round, you get another chance to redeem or be healthy. Whereas males, even 1 round of bad luck means a trait or the physical characteristic is disease. So now do you understand why males are much more likely to get sex-linked recessive conditions, right? The reason is they have no opportunity to mask the recessive disease allele. Okay, so maybe I'll just write that down for you as well. Actually, it's written right here for you. I've written all of this. So females, since they carry 2 alleles, have the opportunity to mask a diseased allele. Mask is a buzzword I want to see in your answer. Okay? If you don't mention masking alleles, We're not explaining sex linkage. So two things I would always want to see in your exam answer is explaining that females carry two alleles, males carry one, and that females can mask their condition. Good. That's the crux of sex-linked inheritance so far. Okay, so these 2 points that I mentioned. Point 1, point 2. You need to have both in your answer. Bookman question?
Yeah, so the bottom it says males are more likely to be affected by sex linkage recessive, but what about dominance? Is it the dominant is the same
Very
thing?
good. I was about to ask you all the same question right after this. So, if you're sex-linked, if the condition is sex-linked dominant, which is very rare, I only know a handful of conditions. One is hypertrichotism, which means being hairy, right? That's a sex-linked dominant condition. Now, who would you expect that to occur? More likely to occur in males, females? Why? Why is that?
Because they only have that one X chromosome.
So we said, so is the answer males? For a sex-linked dominant condition, we're saying males are more likely to get it? Is that a final answer?
No, females.
Females are more likely to get it? Is it equal? Are you sure?
No, because females,
I definitely think it's not equal, but
females, they have that option of also getting dominant.
let's apply the same thought experiment, right? Sex-linked means females can have how many alleles? Males can have? Okay, so that's where we start off, right? Now, if it's dominant, which allele is disease? Capital R or little r? Good. Now, let's think about this. Let's say a female were to inherit a recessive allele, right? Does she have disease? No. But is she healthy yet? No. Because she has to play the disease lottery twice. And then, if she gets capital R, what does that mean? Has a disease. Whereas a male only has to play this bad luck lottery once. If he gets a little r once, he's safe. He doesn't need to play the bad or disease lottery twice. Does that make sense to everyone?
Yes.
So with a sex-linked dominant condition, we'll see disproportionately females be affected, right? Because even if they inherit one healthy allele, they still have a chance of inheriting the diseased allele. Whereas males inherit one healthy allele, they're safe. Okay? The only sad thing is that sex-linked dominant conditions are very rare. So if you ever see the word sex-linked in HSC, by 99% of the time, sex-linked recessive is what they're talking about. Okay? Good. Any questions? So to answer your question, Lookman, it's the exact opposite. If it's sex-linked dominant, you will see males being affected. Sorry, not males, females being affected, way more than males. So females affected more than males. Okay, but this is rare. I've actually never ever, maybe once in the past 6 years, seen a HSC or trial question about sex-linked dominance. So I'd say very low yield, but have it in the back of your mind for your trial exams. Okay. Any questions so far? Good. Right, let's start possibly talking about Plant Squares. Now, this is a checkpoint, and again, I want you to do this for homework, but then again, what I would like you all to do, what you ideally want to do next year, is you want to come into class, you have access to these notes, right? Finish reading through all the notes, self-teach yourself the rest of this module, and do all the HSC Biology additional questions for Mod 5. That way, you're ahead of what we do in class, and if you can all do this collectively, I can go through Mod 5 very quickly, and it'll help you because you will all be even more accelerated beyond your peers. That's something that you all need to take the responsibility of, okay? So maybe write this down, a note to yourself, finish the module, read the theory notes, do all the HSC Biology trial questions for Mod 5. You all know where to access that, right? Do you all have it, is it printed in? It's not printed in your booklet, but it should be online for you all to access on the website. So splash out all of those questions and start Module 6 in your own time. That's what I did, what I recall over my holidays in Year 12. Good. Shall we start looking at pedigrees? Oh, actually, no, we need to go through one final thing, which is incomplete dominance. which I'll take you through. Where is that? Incomplete. Here. Okay. Now, incomplete dominance is very, very simple. It is what the scientific community thought at the time of Mendel, right? Where if you have a tall organism and a short organism, you get a blending of characteristics in a middle height organism, right? So there are two alleles which are both recessive. That's how you think about this, okay? So when they combine together, you get a blending of traits. So here you see, we denote it the same way. We subscript it. So we use C for color, we use R, and you can see here, there's two different letters, but you should just be writing white. It should be like this, okay? Because both are recessive per se, okay? And what we do is, if we have a heterozygote, as you can see here, it's RW, we get a pink organism, right? A blending of red and white gives you a pink phenotype. So the plant that does this is called the snapdragon. This is just one rare example. I don't know any other examples in biology of any species where there is incomplete dominance, so it's quite a famous one. But good. Now, I just wanted to note here, do you all see how it's capital R and capital W? That does not mean it's dominant. It's just the way you subscript it. You can use a little r if you'd like. It's completely fine. You'll not lose any marks at all. Okay? Now, final question. How many phenotypes do we get with incomplete dominance? How many different colors do you see here? Ask a kindergarten kid. How many colors do you see? Already said. 3. The answer is 3. There's 3 phenotypes. So there's red, white, and pink. Do you see how that's similar to codominance? Except the third phenotype in codominance is not a blending, it's patchwork, as you saw with blood type and the Roman count. Very good. Any questions about incomplete dominance? This is probably one of the most low-yield topics in MOFA. I would have it at the very back of my mind. Okay. Good. Let's start doing a question. Let's try. We'll do question 3 first. This is quite an easy one. Now, my question to all of you. Do we do a Punnett Square or do we do a pedigree? So, let's go with Georgina. What are you doing? Pedigree or Punnett Square? For this question here. I'll let you all have a read of this. And Georgina, when you've had a read, you tell us what you think.
Punnett Square.
Good. How did you know to do a Punnett Square and not a pedigree?
Because I thought I would do Punnett Square because there's heterozygous for mother and father, I think.
So the reason I would do a Punnett Square is because I know the inheritance pattern. Do you all see that? With a pedigree, the question is always going to be, what is the inheritance pattern? Okay? So in a Punnett Square, you know the inheritance pattern, number one, and you've been asked about the children or the offspring. If you are ever asked about the offspring, you must do a Punnett Square. Okay, good. So I'm not going to say anything. I'm sure you've all done some semblance of a Punnett Square before in high school. Use that knowledge, and we'll go through how to understand and interpret this. This is quite a very straightforward question. Don't worry about the disease. You don't know anything about it, just the inheritance. Kirthana, I want to hear your answer on what percentage of children will have the disease when you're ready. Remember what we did, right? Draw the genotype of both parents, make a cross, look left, look up, write what you see. And that's the genotypes, and then from that, think about what the phenotype would be. So you should finish about now. It's quite a quick one. Kirtan, are you done? I'll come back to you. Sashida, how are you going? So what should I do now? Tell me. Good, because they're heterozygous for both. Good, good. And then just cross it, and look left, look up, left up, left up, left up. So remember, these are only genotypes. So from the genotypes, you can see what percentage are heterozygous. 75? Good. So if they ever ask you for the genotype, you say 50% are heterozygous, 25% are homozygous dominant, 25% are homozygous. But what if they ask you about phenotype? That's what they're asking here. What percentage will have this disease? Now, if it's autosomal recessive, is it capital R or little r, which is disease? So write it down. Make sure you're at little r equals disease. So what that means is a person will only express disease when they get how many little r's? 2. One's not enough. It would be masked, right? So what percentage have 2 little r's? Well, in this case, I wrote t, so I should have written little t. Yeah, it's only 25%. So your answer is easy. Not too hard, right? Good. Happy to move on? Punnett Square is very easy. It's not a hard part of Mod 5. Question 2. This is a bit more of a challenging question. Not really, but you'll all be fine. Game 2 minutes. How are we going? So when you finish, put your pen down, Simon. And leisure students, raise your hand if you finished. Good. Christian, maybe you can start us off. Okay, so let's write male up and let's write female here. Okay, so what's the male's general chromosome? Is it XY or XX? Good, so write XY. And the male has Duchenne muscular dystrophy. So what letter will they have? Very good. So I think the second we see sex-linked recessive, we should write lowercase r equals disease. I always do that. Always. So I know that this male must have a little r. That's why it's disease. Good. And the female pronoun is going to be what 2 chromosomes? Good. So she can have 2 alleles. Now, what is the female's phenotype? Okay. Pause for a moment. This is where you have to read between the lines. She's unaffected. Does that mean she's capital R, capital R? Does it necessarily have to mean she's capital R, capital R? What could she be? Yeah, or an equally likely situation is the exact same where she is capital R, little r. Equally likely. Now, in the actual HSC, they'll always tell you if she's a carrier, which is what you see here, where she carries the diseased allele, but she's not expressing it, or that she's completely unaffected. I've made this harder for you by not telling you. So you need to weigh up both possibilities. Okay. Good. All right. So do the Punnett Square for me now. So what should we get? So when you fill this in, you'll get x capital R, x little r, x capital R, y, x capital R, x little r, x capital R, y, capital R, x little r, x capital R, y, x little r, x little r, and x little r, y. Okay, so what proportion, now this is where we have to read the question really hard. Is it asking what percentage of people? Is it asking what percentage of children, elderly, males, females? What is it asking? Very important. So do we care about any of the XYs? So our sample space or our denominator is the female. So what we can do straight away is cross out all the males. Like that. Okay? So the question is, what percentage of females will have Duchenne muscular dystrophy? Now let's look at the first case, case 1. What percentage have Duchenne muscular dystrophy, everyone? None of them. Both of them have the dominant allele to mask the disease allele, so 0%. What about in case 2? 80%. Now, remember, we don't know if this is a case or this is a case. So what should we do? These are 2 independent events. Female can either be this or either be that. Now, basic probability, what do you do with the probability of 2 independent events? times them together. So what are you going to do with this case? I'll find the average is what you would do, right? Because timesing by 0 is 0, right? So the average probability is going to be, overall, what would you all say? 25%. So the challenging part of this question was this part, which I intentionally left very ambiguous, so you took both possibilities. Now, in the HSC and in trials, I'll typically always give you one or the other. But hopefully you've done now the hardest version of a Punnett Square that can be. Okay. Any questions? I think, Lookman, did you have a question earlier?
No.
Okay. Does anyone have any questions? Does that make sense? Yeah? Georgina?
How did you get 25%?
Yeah, so I just averaged 0 and 50. So I added the two percentages, divided them by 2. So I found the average percent likelihood. That was 0% plus 50 and then divided by 2. Yeah, very good. Lookman?
Yeah, couldn't you just look at the other one and see that there's only 25% of females there? Like, how did you get 50% for the second one? I just got 25% of females in the second Punnett Square.
Okay, now remember the question is what proportion of females will have D&D? So would you include males in your sample space?
Oh, yeah, okay, I understand, yep.
Yeah, so the second they say what proportion of females or males, you need to narrow your sample space to females or males, and then within that, the numerator should be how much are affected. Good. All right, well, that concludes this year, right, for bio. So you've all learned up to Punnett Squares, which is very good. What I would like you to look up on right after this is pedigrees. And I'd like you to take it all the way to the end where we're very close to finishing, to be honest. It's about 30 pages of theory for you to read on your own, which is SNPs, PCR, gene sequencing, profiling, and then the top point would be about health, human evolution, and species conservation. So you're done after that. Okay, so look into that, do all the additional questions. I'll still always be a message away if you have any questions at all. And good luck. Make sure you all have a system to do work. So what are you all going to do to make sure you get work done in the holiday? I'm telling you, there'll be days where you just feel like not doing anything, right? And that's normal. So what systems do you all have in place to do work on the days you don't want to work? Okay, one thing I would say is, yeah, you could like do Pomodoro, but have breaks in sessions. Yeah, but what else could you do to force you to do work? Fun? Yeah, you could have rewards. Again, for me, it doesn't really work. I just take the reward and I don't do the work. So, what could you do?
Group study set.
Yeah, group study session, right? Say, you know, tomorrow I'm going to teach you gene sequencing, you're going to teach me profiling. And if you rock up not knowing anything, you look bad. So you don't want to look bad, right? So that puts your personal accountability at stake. What else?
Make flashcards, and if you don't feel like doing any like hard study, just grab flashcards. Yeah,
you could find more passive ways to work as well. So flashcards, so you can just look at past recordings of lessons and just, you know, not really think about it too much. Anything else you can do? I think the hardest thing, the thing you should all focus on is how can I externalize accountability? How can I take my accountability and swap it with someone else or throw it on someone else? So try finding maybe one study buddy that's similar level to you, that can challenge you, and you can swap accountability in the sense that maybe you can message each other what you did in the day, or you can study together quietly on a Zoom call and make sure you all attend. Those are small ways you can do it. So I want you all to realize that HSC is not about how motivated you are. I think all of you have had phases where you've been like, I'm going to smash it. I'm going to work as hard as I can. And a week later, you're like, I can't be bothered, right? And the same with me. But the people that are truly successful are those who can really push through in the days that you don't want to get work done. So that's what I want you all to focus on. Okay? Good luck. You got this. And I will see you all